Hello, welcome back everyone. This lecture is going to be covering the second part of chapter 2 from your Marab text, which is covering chemistry. In this section, we are going to be talking about biochemistry. Okay, and biochemistry is the study of chemical composition and reactions of living matter. All right. So all compounds are either going to be classified as organic or inorganic. Inorganic compounds are inorganic because they do not contain carbon, whereas organic compounds do contain carbon. Okay, so some examples of inorganic compounds, you have water, salts, and many acids and bases, which we'll talk about a little bit more in a second. Organic compounds are, again, anything that contains carbon, so carbohydrates, fats, proteins, and nucleic acids. And they're usually going to be large and covalently bonded. And now remember, we just talked about covalent bonds. Covalent bonds is when two atoms share their electrons, right, to try to make their valence shell full. So carbon has 6 electrons total. So the first shell holds 2. The second shell then would only have 4. So carbon wants to get 8 electrons in its valence shell. So it's going to want to share 4 electrons with another molecule or atom. Okay, so carbon is going to be covalently bonded, and it's going to make organic compounds. All right, so let's start with some inorganic compounds. So at the top here, and the rest of the slides, I just noted if you're talking about inorganic or organic, just to keep everything straight. So the first inorganic compound without carbon that we're going to be talking about is water. This is the most abundant inorganic compound, and it accounts for about 60 to 80% of the volume of all living cells. I don't know if you've seen the memes where it just says we're all just sad cucumbers because we're all just water with feelings. But the majority of the human body is made up of water. Okay, and it's really important because of some specific properties, and I'll go through each of these in detail. But it has high heat capacity, high heat of vaporization, polar solvent properties, reactivity, as well as cushioning. Okay, so what does all that mean? High heat capacity is the ability to absorb and release heat with little temperature change. So water is really good because it prevents sudden changes in temperature. Okay, so our body is mostly water, and because of that, if we step outside in the sun, we don't immediately get hot, right? The water in our body prevents our body from changing temperature suddenly, which obviously we don't want. We already talked about homeostasis and how we want to maintain normal body temperature, which is 37 degrees Celsius. Water is what allows our body to maintain that to some extent, right? And then we have other physiological processes that we can use to warm up or cool off our body if it gets too far out of that range. Okay, the second property, high heat of vaporization. Evaporation requires a large amount of heat, okay, and it's useful for a cooling mechanism. So again, really important in maintaining our homeostasis with our internal body temperature. The water's ability to evaporate and use up large amounts of heat it allows our bodies to be cooled off more efficiently. Okay. The next property is the polar solvent property. So over here on the right, we have a drawing of the water molecule. What type of bond is water bonded by? The hydrogen and oxygens. Okay, you should know it's covalent. Is it polar or non-polar? You should know that it is polar. Okay, we already talked about that. If you're not sure what that means, go back and review our part 1 of this lecture. But water molecules are polar covalent bonds, which means they create a dipole. Okay, a dipole is a partial separation of charge. So you have one side that's partially positive, where the hydrogens are, and one side that's partially negative. okay, over by the oxygen. This means that if you use water as the solvent, the liquid that's going to dissolve the solute in some sort of solution, that means if you place an ionic compound, so a substance that has ionic bonds into water, where you have this dipole, it's going to be able to dissociate the ionic compounds, which means if you have sodium chloride together, Na and Cl, when you place that into water, sodium is a positive ion, cation. Chloride is going to be a negative ion, an anion. When you put that in with a water molecule, since the sodium is positively charged, the partial negative on the oxygen molecules is going to want to bond to it. So this is our poorly drawn water, right? Our oxygen molecules that are partially negative are going to be attracted to the sodium. So you can see here, the negative oxygen regions of the polar water molecules are attracted to the sodium cations, whereas the chloride anions is going to be the opposite. So the hydrogen, the two blue parts here is what I'm drawing these circles, those positively charged, partially positively charged hydrogens are going to be attracted to the chloride. So what's going to happen is these ionic compounds, so sodium chloride, are going to dissociate, which means they split up into their individual parts when they're placed in water. Okay, and we'll talk about this a lot more in lab and in some upcoming chapters where we talk about diffusion and osmosis. But it's really important to know that ionic bonds dissociate when put into water. Okay, and this is another reason why water is so important, because it allows us to transport a lot of different electrolytes and salts through our body, which is really important to keep our body moving. Okay. Then two more properties of water. Reactivity, it's a necessary part of hydrolysis and dehydration synthesis reactions, which we're not really going to talk about in this class too much, just because it's a bit over our heads, but it's important in a lot of chemical reactions. You need water for some chemical reactions to happen. And then finally, cushioning, which is very important. Water is going to help protect certain organs from physical trauma. So your cerebrospinal fluid is a watery fluid that surrounds your brain and spinal cord, and it helps cushion it. So to make sure that if you get hit in the head, hopefully there's enough fluid there to cushion your brain so that it doesn't cause physical damage to your nervous tissue. Obviously, if you get too hard of a hit, it's going to damage the tissue, but the fluid acts as a very good cushion. Okay, so those are all physical properties of water and why it's such an important inorganic molecule. The next inorganic molecule are salts, which you should already know are ionic compounds, right? Ionic bonds are bonds, remember, where the electrons are transferred, right? So, for example, our sodium chloride, right? Sodium loses an electron, so it becomes partially positive charge, and chloride gains an electron, so it becomes more negatively charged. And again, remember, we're transferring electrons to try to make our valence shells either full or get rid of extra electrons we have to move down a valence shell step. Okay, and we talked about already that salts dissociate into their separate ions in water. So sodium and chloride, when placed in water, are going to separate and be individual. And separate into cations, which are the positively charged molecules, and anions, which are negatively charged. Okay, we've already discussed that. Ions are called electrolytes because they can conduct electrical currents in solution. And it's really important for our bodies to have these different electrolytes or salts or ions floating around in our fluids. Helps with a lot of physiological processes. Well, it says it right here. Ions play specialized roles in body functions. So sodium, potassium, calcium, and iron specifically, really important when we talk about muscles and how they contract, the physiology behind muscle contractions, or when we talk about action potentials being transmitted through the nervous system. You will see all of these ions play a major, major role. And if we don't have those ions present, our body can't function like it's supposed to. Okay, also very important in homeostasis. And then just some common salts. You don't have to memorize those, but I included them. I don't know if you wanted to know. Okay. Then the next two inorganic molecules we're talking about are acids and bases, which I'm going to try to simplify a little bit because we'll talk about this more later on. But very basically, acids are proton donors, which means they release hydrogen ions. Okay, hydrogen ions are positively charged, so is that a cation or an anion? Cation. Bear protons into solution. Bases are the opposite, so they're proton acceptors. So they are going to pick up spare hydrogen ions in solution. Okay. When a base dissolves, it releases a hydroxyl ion, OH negative. That's going to pick up the H positive. So important acids listed there, important bases. You'll see this a lot in BI-208 when you talk about the respiratory system and your acid and base balances in the blood, gas exchange, and stuff like that. Okay, we can measure acid-base with the pH scale. So it's a measurement of concentration of hydrogen ions in a solution. Acidic solutions have high concentrations of hydrogen ions, but the pH is low. So let's see on here. These low pHs are acidic. Okay, which means that they have more concentration of hydrogen ions. Neutral solutions fall in the middle, around a pH of 7 there. They have equal numbers of hydrogen and OH minus. Pure water is neutral. And then the last one is your basic solutions or alkaline, same thing. They have low concentrations of hydrogen and a high pH. Okay, so on the scale above 7, this would be your basic solutions there. And there's some examples here. So basic solutions, different oven cleaners, different household cleaners, ammonia, bleach, egg white is basic, blood is a little bit basic. Acidic solutions, obviously some of them are kind of straightforward. So things like lemon juice, coffee, wine, and obviously like strong acids that you would see in lab. Okay, so that's acid and bases. So those are inorganic substances. Now we're going to be talking about organic. And again, things that have carbon in them are considered organic. Okay. Carbon is considered electroneutral, which means that it shares electrons. It's never going to gain or lose them. So you're never going to see carbon in any sort of ionic bond. Instead, it's always going to want to form 4 blank bonds with other elements. Can anyone tell me what's supposed to be in there? Anyone answer? It would be 4 covalent bonds. Okay, it's never gaining or losing your electrons. It's just going to share. And sharing electrons is what forms covalent bonds. Okay, so some organic compounds we're going to talk about. Carbohydrates, lipids, proteins, and nucleic acids. Most of these are what we consider polymers, which are long chains of smaller subunits that are called monomers. Okay, so monomers are like individual small subunits. Say this is a monomer, this is a monomer, this is a monomer. If they all get combined together to form like a long chain, say these are attached now, that whole thing's a polymer. right? So a polymer is made up of individual monomers. Okay, so all carbohydrates, lipids, proteins, and nucleic acids, those are all examples of polymers. So the monomers are going to be the building blocks we're going to talk about that make up those larger units. Okay, so let's start with carbohydrates. Again, organic because it contains carbon. Sugars and starches are examples of carbohydrates. 3 classes of them: monosaccharides, which is just one simple sugar, which is just another name for a monomer of a carbohydrate. We have disaccharides, which are 2 sugars together, and polysaccharides, which is many sugars. Again, polymer is another name for a polysaccharide, right? So if I was, each circle is a monomer, so each circle is a monosaccharide. For a disaccharide, we'd have 2 monosaccharides or 2 monomers together. In a polysaccharide, you'd have many monomers or monosaccharides in a long chain. Okay. So here's some common examples of monosaccharides first. Again, carbohydrates are just sugars. So you have glucose, fructose, galactose, deoxyribose, and ribose. Again, you can see all of them. Let me get a brighter color. All of them are just this one monomer, right? It's just a single carbon ring. Disaccharides, sucrose, maltose, lactose. You can see each one of these is made up of 2 individual monomers. So sucrose is glucose plus fructose. Maltose is 2 glucose molecules together, right? So disaccharides have 2 monomers. And then polysaccharides are long chains. So for example, glycogen, you can see this whole thing is just a long chain of monomers that have been added together, and they form these different long shapes. Okay, so that's a polysaccharide. The next class of organic molecules are lipids, which are just fats. They're insoluble in water. And there's 3 main types, which we'll talk about a little bit later on. Your book has this table here. I'd recommend you go through and take a look at those. Some more information that I may not talk about, but is important. So first, triglycerides are called fats when they're solid and oils when they're liquid. Okay, so lipids are the name's kind of interchangeable with fat. But when they're solid, they're considered fats. When they're liquid, they're considered oils. Triglycerides are composed of 3 fatty acids bonded to a glycerol. So our fatty acids are going to be these 3 fatty acids here. Right? So those are our fatty acids bonded to, you're not going to be able to see any color. I'll do gray. Bonded to this glycerol molecule up top. Okay, that's the general makeup of a triglyceride. Main functions, think of, what you think of fat being the function in your body. Energy storage, insulation, and protection. So help keep your body at a normal body temperature, store energy for later use, and then adds that fatty cushion to help protect deeper tissues. These fatty acid tails here, that there's 3 of them in the triglyceride, they can be in 2 different forms. Okay, so you have either saturated or unsaturated fatty acids. Saturated fatty acids, all carbons are linked via a single covalent bond. Okay, which means you have the maximum number of hydrogen atoms possible. It's saturated. Right? Saturated with hydrogen. These create linear molecules which can pack closely together, forming a solid at room temperature. I'm going to do that a different color. Solid at room temperature. Okay? So any fat that's solid at room temperature, different animal fats, butter, is considered a saturated fatty acid, again, because you have the maximum number of hydrogen molecules possible. And I'll show you an image of that in a second. The other type, unsaturated fatty acids, some of the carbons are linked by double bonds, which means you don't have the full amount of hydrogen. Okay, the double bonds causes kinks, so it can't pack closely together. So unsaturated fatty acids are liquid at room temperature. Okay, so plant oils such as olive oil, avocado oil, canola oil, a lot of things, oils you see like in cooking, those are all considered unsaturated. Okay, and two more specific examples, which we're not really going to talk about at all in 207, but you'll kind of get when we talk about digestive system in 208, trans fats and omega-3 fatty acids. Just kind of interesting, but you don't have to know those. Okay, so here's what saturated fatty acids look like. Again, saturated because you have a straight line, right? And you have as many hydrogens attached as possible. Again, these are going to be solid at room temp. Whereas unsaturated, we're going to have a double bonds at some points, which is going to cause a kink in our chain here, which is going to make this not pack closely together, which means it's going to be liquid at room temp. Okay. Phospholipids is a specific type of lipid you may have heard about before. It's a modified type of a triglyceride, which it just has the glycerol and 2 fatty acids instead of 3. But it also has a phosphorus head group. So this phosphorus head group. So that ends up giving us a head and the tail region. The tail region is these fatty acids. Fats are going to be hydrophobic. The head group is polar. Water is polar. So it's going to be hydrophilic, which means it loves water. Okay. So you're going to see these phospholipids make up our cell membrane, which we'll talk more about in chapter 3. But they form this bilayer. So you have the polar heads on the outside. So say this is the outside of the cell. So you have water outside the cell. You have the fatty tails along with the other fatty tails, creating this semi-permeable membrane. And then the polar heads on the inside of the cell, where you'd have the cytosol, right, the internal contents of the cell. Okay, that's the phospholipid bilayer because there's 2 layers. The last type of fats are considered proteins. So these are 4 interlocking ring structures. Most important one that we're going to talk about is cholesterol. Starting material for synthesis of vitamin D, steroid hormones, and bile salts. So a lot of things in our body are made of steroids. It's really important in the cell plasma membrane structure, which again we'll talk about more in the next chapter. So this is what a typical steroid looks like. You'll just see these 4 ring structures. And depending on this group attached to the ring structures, it'll be a different type of steroid. Okay, but this is the basic look of a steroid. Okay, next organic molecule we're going to talk about are proteins, which comprise 20 to 30% of our cell's mass, have such varied functions. There's proteins for everything, do everything, structural proteins, functional proteins, chemicals that are proteins. The polymers of amino acids are held together by peptide bonds. So the monomers in this case are the amino acids, right? So the individual building blocks are going to be amino acids. So say we have a bunch of amino acids here, right? They're going to be held together by peptide bonds, right? So that's what makes up our proteins. Again, our monomers are the individual amino acids. The shape and function is due to 4 structural levels, which we'll just briefly touch on. Here's the different types of proteins you can have. I'm not going to worry about this too much right now because later on in the semester, we'll talk about all of these different things in detail. So just know that proteins have a wide variety of functions. As long as you know that, you should be pretty good. So again, amino acids are the monomers, the building blocks of your proteins. And again, they're joined by the peptide bond here in the middle. Okay, I'm not going to ask all these fancy questions about the amino acids and stuff. Just know amino acids are the monomers held together by the peptide bond. And then the structure of protein, there's 4 different levels of them. So we have primary, secondary, tertiary, and quantitary. I'll show you images of these, I think. You can pause and write this down if you want this in your notes, but I think the pictures kind of make it make a little bit more sense. Okay, so the first is the primary structure, which is just a sequence of amino acids in the chain, right? So here are our individual amino acids all lined up. Again, the amino acids are our monomers. So just the sequence in the single chain is the primary structure. Okay, this primary structure is going to fold up and create more complex shapes, and that's considered the secondary structure. So we can either form alpha helices or beta sheets. Okay, so helices, this primary structure is going to coil, right, and form a helix shape, and it's held together by hydrogen bonds. So you can see, right, if the blue is hydrogen and we have oxygen, between the hydrogens and oxygens, you form these hydrogen bonds that are going to keep this in its spiral shape, right? These hydrogens and oxygens on the original primary structure, as that folds up, it's going to create this helix shape. Or you can have these beta sheets, which is similarly held together by hydrogen bonds as well, between, again, the oxygen and the hydrogen molecules. Okay. Oops. Then we have our tertiary and quaternary structure. So tertiary is just superimposed. So you have helices or beta sheets are folded up even further to kind of form a globular molecule. Again, there's going to be bonds that hold all this together, but you're going to have alpha helices, different beta sheets all fold together to make a tertiary structure. And then quaternary structure is going to be 2 or more of the chains, each with its own tertiary structure, formed together to form a functional protein. Okay, so you're going to have multiple tertiary structures coming together to form a big functional protein. Okay, as long as you know the 4 different types of structure and just very generally how they're made, you'll be okay. The shapes of the proteins you can differentiate into 2 categories. The first is fibrous, which is structural proteins, so they're not functional. These proteins are strand-like. They provide mechanical support and tensile strength. So things that are in our body that are just there to help hold everything together. So keratin, elastin, collagen, the different proteins that we find in our skin and in our connective tissue that are there just to help make our body shape what it is are fibrous or structural proteins. We also have globular or functional proteins, which are the proteins that are actually going to be doing the shit, right? So they're going to be compact, spherical, water-soluble, sensitive to changes. They're going to be things such as antibody, hormones, enzymes. You have different protein channels, things that are actually going to serve some sort of functional purpose in our body. Enzymes help digest, catalyze reactions, whatever, right? So these functional proteins are what's actually going to be doing the work. Okay, and those functional proteins can be subject to what we call denaturation, which is when those proteins unfold and lose their shape. So the only reason they're able to do the job that they're supposed to do is because they're in their specific shape. And we'll talk about it a little bit more later on, but that shape allows them to bind to other molecules, other chemicals, and do their specific job. So if they're in a situation where they have decreased pH, an acidic environment, or increased temperature, it can denature the protein, okay, which makes it no longer functional. So an example of this, in your saliva, you have a protein, an enzyme specifically called salivary amylase, which is important in digestion of starch or the polysaccharides. So when you're chewing a cracker in your mouth, if you leave it in your mouth long enough, you'll start being digested by this amylase. As soon as you swallow that food and it enters your stomach has a lot of HCl in it. That decreased pH is going to denature the amylase, right? So starch digestion will stop. Once the starch and amylase hit your stomach, no more digestion is going to happen because it's too acidic of an environment. Okay, so that's just one example. If it's not super severe of a denaturation, it can be reversible, but in extreme changes, it's irreversible. So cooking an egg is an example of denaturing that egg's proteins. Obviously, once you cook something, you can't uncook it. So in very extreme situations, it can't be reversed. Okay, enzymes like the amylase we just talked about are examples of globular or functional proteins that act as catalysts. They increase the speed of chemical reactions, and they don't get used up within the process. So you can have salivary amylase, a little jar of it, and it'll continue to break down starches over and over and over again, and you don't need necessarily to create more amylase as long as yours is still good. Enzymes are specific. We'll talk about this again more later on, but each enzyme, the way the protein is folded into its tertiary structure, it can only act on a specific substrate. Okay, so amylase, digests starch or polysaccharides. We have enzyme protein called lipase that digests lipids. Okay, amylase can't digest lipids because the shape of the enzyme only allows it to bind to a specific substrate, which in that case would be starch. Okay, often the names end in ase, amylase, lipase. So anytime you see that, it's a good indicator it's an enzyme, and it's often named for the reaction that they catalyze. Okay. Again, this is kind of reiterating what I already said, but it lowers the activation energy, which is the energy needed to initiate a chemical reaction. So for example, breaking down a polysaccharide into monosaccharides, digesting starches. Without the enzyme, it's going to take a lot of energy for that to happen. With the enzyme, it's going to happen a lot more efficiently. So it's going to reduce the amount of energy needed and allow it to happen more quickly at body temperatures. Okay. Three steps in enzyme action. This is pretty straightforward, but first your substrates bind to the enzyme, right? The complex undergoes rearrangement of the substrate, so the enzyme does whatever the enzyme is doing, okay? And then the product is released from the enzyme, okay? And then this enzyme, again, doesn't get used up in the process, so this enzyme can go on and continue to do this work with other substrates, okay? Our last type of organic molecule are nucleic acids. They are the largest molecules in the body. Their monomers are called nucleotides. Okay, so your nucleotide monomers, right, we talked about before. bound together make up nucleic acids. Okay, and there's two main types of nucleic acids, which these hopefully are familiar with you. Deoxyribonucleic acid, and then just ribonucleic acid, which, if you don't know what those are, DNA and RNA. Okay, so DNA holds a genetic blueprint for the synthesis of all proteins. We'll talk about this more in the next chapter. It has a double-stranded helical molecule. Again, the nucleotides contain deoxyribose sugar, a phosphate group, and nitrogen bases, adenine, guanine, cytosine, and thymine. And the bonding of these bases are very specific. So A always pairs with T, G always pairs with C. Okay, and don't stress out too much about this. This is in an intro biology class, so you don't need to memorize everything here. But again, you have the sugar phosphate backbone, so these purple spirals. And then the rungs are the nucleotide bases. Okay, and they're going to pair together and form this double helix. So that's DNA. RNA is very similar, but it's just a single-stranded molecule. Contains ribose sugar instead of deoxyribose. The base thymine is replaced with uracil. There's different types of RNA. Again, don't stress out too much about this stuff. I'm really not super worried. Okay, so here's the list of monomers and polymers we've talked about. So for carbohydrates, your monomer is a monosaccharide. Polymer was the polysaccharides. Proteins are monomers with amino acids. Polymers were the proteins, or polypeptides. And then nucleic acids, monomers were nucleotides. And then our polymer would be either the DNA or the RNA. Okay. And then the very last bit here, really quick, I just want to mention ATP, adenosine triphosphate. That is where our body is going to get energy. So ATP directly powers chemical reactions in our cells. It offers immediate usable energy. And you have, I'm not worried about the structure really here, but you have the adenine containing RNA nucleotide with 2 phosphate groups. And we'll see this throughout the rest of the semester in 207, that ATP is needed in a lot of different processes in our body. So transporting materials through facilitated diffusion, which we'll talk about in the next chapter, mechanical work such as contraction of muscles, and chemical work, so driving and absorbing chemical reactions in our body. Okay, so that is the end of chapter 2. Let me know if you have any questions.